Hello, I'm Michael Schrader, an upcoming fifth-year biomedical engineering student, and this is my presentation for the RIT Undergraduate Research Symposium in collaboration with Dr. Michael Richards on the subject of ultrasound imaging metrics of live and cadaveric human flexor tendons. The motivations for this research include the frequency of these injuries. Many thousands occur every single year, and the fact that these injuries typically result in surgery, even if physical therapy is used as a first measure. Despite the frequency of these surgeries, the post-operative protocol isn't standard, and that results in variability for the outcomes on this physical therapy, and often recurrent injuries and surgeries. This is really important because each of these lacerations in the tendons results in upwards of $70,000. Onto some clinical background. The flexor tendons run from the fingertip down through the wrist into the forearm muscles, and the injury is typically caused either from a laceration along the palm or an avulsion where a closed hand is forced open. Think of a rock climber losing their grip and the tendon pulling off of the bone. These injuries commonly result in surgery because anything beyond a sprain is going to need to be surgically repaired. Despite the frequency of the surgeries, the typical physical therapy window is quite variable. As an example, you'll have 8 weeks of inactivity, followed by 4 weeks of manually bending the fingers with the other hand. Then weeks 4 through 7 spent with wrist motion with relaxed fingers. And then after that, about 5 weeks of simple finger flexion exercises as demonstrated in the bottom right. So a possible cause for this variability is the changes in an individual's anatomy, which result in different patient-specific forces in that person's tendon. To explore the project overview, our hypothesis is that elastographic metrics estimated using a standard clinical ultrasound imaging system may be correlated to forces in the tendon while undergoing rehabilitation exercises. In order to prove this hypothesis, we have to measure total strain and shear wave speed along a proximal portion of the tendon during exercise using longitudinal ultrasound imaging sequences. We will then correlate those ultrasound metrics to an independently measured force from the tendon. We will validate these techniques in both cadaveric studies and in vivo studies. In the cadaveric studies, we will measure force from the fingertip and from the flexor tendons directly. And in the in vivo clinical studies, we'll measure it just from the fingertip. The purpose of this work is to develop the experimental setup and exercise protocol that will be used to perform these validations. For the general experimental protocol, what will need to be done is measuring force in the finger using a force load, to collect longitudinally aligned ultrasound images, meaning aligned with the finger, and then to collect quasi-static B-mode and shear wave images using the ultrasound. The B-mode images will be used as an input for a custom image registration algorithm to measure total strain accumulated during the exercise. To collect these images, the hand will be submerged in an acoustic coupling solution, which is used to maximize the image quality. For the cadaveric protocol, a hand will be mounted to a custom-built frame with 2 6.5mm K-wires in a submerged bath. The finger will be held steady by a loop attached to a force sensor at the fingertip. The flexor tendon will then be attached to the instron from a pulley from the proximal side. The load will then be increased in increments of 10 until it reaches 70% of the failure load, starting at approximately 25 newtons as a preload. This will be repeated on each finger. To continue the cadaveric experimental protocol, a laceration will be made along the base of the finger in zone 2 of the hand and will be repaired using a cruciate configuration suture, which is consistent with the clinical standard of care used in live patients. The process described in the previous slide will then be repeated on each surgically repaired flexor tendon until there's a failure in the suture. As for the experimental protocol in the clinical subject, this will serve to validate the overall methodology used, as the force applied from a muscle may be different from the force applied from an instron. This will be tested by using 2 exercises with a video camera recording at the side to measure the angle of flexion. The first will be a simple range of motion with no force load, and the second will be an isometric with a force load attached to the fingertip. To begin the experimental design, we'll start with the transducer holder arm, which obviously will hold the transducer probe in place. Some requirements for this is that it must be adjustable enough to capture a small human flexor tendon. It must be stable enough to allow for repeated capturing of an ultrasound image sequence. We started with arm 1, a repurposed flexible smartphone mount. It was capable of some of these requirements, but it wasn't ideal. It was too loose and it was elastic, which did not allow for fine adjustments in the position of the ultrasound probe. The second arm was an adjustable clamp articulated camera arm, which was similarly flexible, but due to the dials on the side, it could be made stiffer to hold into place for multiple images. To continue the experimental design, the transducer holder was designed next. Its function would be to hold the transducer probe in place on the transducer holder arm. Some requirements include 3D printing to allow for an iterative process where changes could be made easily. The probe must be held stable relative to the arm, and then the probe must be held stable relative to the holder. For the original design, it could hold the probe, but couldn't hold it still and couldn't attach to an arm. The second design could attach to the arm, but still really couldn't hold the probe still. The third design could hold the probe still using a Velcro strap and also attach to the first arm. But I also reduced the impact it would have on readings by reducing its overall size. Onto the final design of the transducer holder. It allowed for the greatest control and was stable in all planes of movement, which allowed for the greatest image quality to be gathered. Its smaller size also made it easier to maneuver and limited its impact on the readings. Additionally, it could be affixed to the articulated arm with just a 1/4 inch nut. To continue to the custom Cadaveric hand mount design, the requirements included that it must be stable while still allowing for subtle changes in the location of the hand and the transducer probe. It must not leak water, and it must fix to the instron platform to allow for the most accurate readings. In the final design, these were solved by using 38 threaded holes to allow for moving of the K-wire blocks, as well as K-wire blocks that could be moved vertically and horizontally along the T-slotted aluminum. Here is a picture of the final Cadaveric experimental setup, with arrows pointing to each of the components, including the K-wire blocks and slides, the instron pulley stand, the transducer holder and probe, as well as the transducer holder arm and the instron. Let's continue to the experimental design of the clinical hand mount. Some requirements for this is that it should also be 3D printed to allow for an iterative process, as with the transducer holder. It must hold the subject's hand in place without causing discomfort. It must prevent out-of-plane motion that would ruin ultrasound image sequences, and it has to fit different hand and wrist size. As for some iterative changes made on these designs, the first one was just an angled board to let the string attached to the fingertip to the force load go out of the water. In the second design, the angle of that board was changed to be more comfortable, as well as a wrist strap to hold the wrist in place and a wrist rest to make it more comfortable. In the third design, wrist contours were added, as well as multiple holes to allow for different sized hands to fit, Velcro straps to hold the top of the hand down, as well as raised walls to hold the hand in place horizontally. Here is a picture of the final clinical hand mount within the setup. You can see the clinical hand bath, the transducer holder arm, as well as the transducer holder. Here are some preliminary results and conclusions, which proved to be pretty promising. All the experimental designs and protocols met requirements, and the preliminary data suggests that the ultrasound image sequences can be collected. Here's an example of the cadaveric image sequence. And here's an example of the clinical image sequence with tracking. As for the prospects of future work, the experimental protocol will be validated with future experiments, and the relationship between total accumulated strain, shear wave speed, and force realized in a tendon will be verified using finite element models. These validations for the experimental protocol will include 5 fresh frozen cadaveric hands to be used for in vitro validation, as well as 10 healthy volunteers to be recruited for clinical validation. Thank you for watching this video.